So we heard a lot about what is possible in an ideal world, what technology can be provided, what scientific can provide to the world. But right now we're starting into a session of real world examples. Yeah, I'm introducing Daria Marsh from the Cell and Gene Catapult UK organization. So she will tell you a bit about the business model of Catapult in the UK. I'm very happy that she's here because she's giving us then also a practical example on how to scale ATMPs. Bastian mentioned that a little bit. Yeah, it's not just automation, there are much more things around it, right? So Daria, thanks for being here, and stage is yours.
Thank you. And thank you very much for inviting me here today to this fantastic facility and this fabulous event. I am head of bioprocessing at the Cell and Gene Therapy Catapult. So I'm responsible for our bioprocessing capabilities, which are focused on scaling across cell and gene therapy modalities. For those who haven't heard of the Cell and Gene Therapy Catapult before, we are an independent innovation and technology organization set up by the UK government. And our goal is to promote the delivery of advanced therapies globally. We do this by establishing collaborations because we believe collaboration is the way forward. We have assets, skills, and developments that we bring to those collaborations to help with innovation, and we work with academia, healthcare providers, and other industry organizations within those. So as you have heard in a wonderful previous talk, ATMP demand is still high. Commercially approved ATMP products, though, far outweigh patient demand at the moment. And what's more is that price point is very high. We are now pressured to improve margins, and this is what investors are looking for in the future. Last month at the Alliance of Regenerative Medicine meeting, they discussed this on what will the price point look like and what will the margins drive look like. And they think it is aspiring to 80% from the current 15 by 2030, which is a huge target for us to work towards. Now, it was thought over the years that scalability will be achieved through allogeneic cell therapy. So some of the CAR-T product manufacturers, for example, always had allo programs going as the second generation products so that they could prove safety with the autologous product and then solve scalability through moving to allogeneic products. And allogeneic products have been making great progress with steady trial numbers rising over the years, but they're not there yet in commercial application. So challenge for scaling at the moment means that we have to address automation in both spaces to make sure that we have a steady path towards the future. So I'd like to tell you today about our approach to automation in the scale-up space and for scale-out as well. You will see these are at very different levels of on the automation journey right now. The case I'll talk to you about in scale-up is an iPSC-derived space, and our approach has been simple here, to first close the process, maximize yield and quality within this process closure, and use PAT to automate a control. We are some way on that journey of PAT implementation. And here what's important to say is that iPSC products and cell products in general are highly complex, and this is part of the challenge. Understanding your product quality is an immense challenge here, and then controlling your process again is a challenge yet again, which can be, we believe, requires automation in order to achieve the process consistency, reproducibility, robustness that is needed in the future. I will then talk about the scale out, but that will come a little bit later. So for our scale-up approach for IPSC expansion has been to use a stirred system. Many IPS processes, and classically these have started off in adherent systems in 2D. As bioengineers, we have stirred things for many years. We like to stir things that have highly characterized systems, stirred tanks, and so we didn't overcomplicate here. We have developed an intensified process within a stirred tank reactor, which starts with a single cell suspension. Now these are adherent cells, and so they then form aggregates within that suspension, and those aggregates expand to a maximum size where we can then harvest them. Now, if you're familiar with IPS culture, it is highly laborious because of the media exchanges required. They are daily. IPS scientists often don't have weekends. They feed the cells through the weekends as well. So we needed to automate this, and we've done that by implementing a perfusion device connected to the STR platform. This uses acoustic separation. And in this manner, we are able to now achieve a consistent aggregate size with this automated daily exchange setup. And we have begun to scale it up. We are now at about a 2-liter scale within the system, and look at seeing a very good scalability. The next manual step in the process is the passaging of the cells, which means that when your aggregates get to a certain size, and that size is maximum if they get too big, the core, the middle of the aggregate will start to become necrotic and your cells will be dying. So we need to dissociate those aggregates back into single cells, and we need to reseed and start the expansion again if you want to achieve a certain output. That's also quite a laborious process, and so we have worked to automate this. We are now, call it in a semi-automated state, where we have used centrifugation, counterflow centrifugation to harvest the dissociated cells and seed them back into a bioreactor. And we can achieve consistent performance through reseeding and this closed and semi-automated process. At the small scale, we use a device called the Retia for this, which can be scaled up to a larger scale centrifuge if you're looking at scales above 1 liter. Now, on the analytics side, one of the tasks that's required for during culture is a analysis of the images for aggregate size. As I mentioned, the aggregates cannot get too big. Now, this is typically done through a manual sample on the bioreactor, which then a scientist will take over to a microscope, take some images, but those images then need to be sized. Now, a few years ago, we wrote some software that would do this image analysis for us, but running many images would still take days through many experiments that you might want to analyze. Now we have automated this with a software called Veda. This is a commercially available software that has cut down this time, which was a multi-day analysis time. In fact, I distinctly remember one of the scientists for a deadline having multiple laptops set up just to get through the images that we had to get through for that project. This kind of flow can now be done in about 15 minutes with a fairly limited training data set, which can be fed in at the beginning of the implementation. Now, what would be really nice is to have this data available online and not to have to do the at-line sample. And so we have now moved to this and evaluated the Sabre tool, which is a camera attached to the bioreactor, which can do this monitoring for us now automatically. Now, this move from 2D to 3D process can be quite risky, and amongst therapy developers, it is quite a risk for them to take. They're not sure whether it's going to work, whether it's going to be beneficial, how their cell line is going to perform, what is the product quality going to be afterwards. And so this is a case study that I'd like to present to you with a project we did with a company called Plasticell. They are a UK company who have a combinatorial screening platform for development of differentiation protocols. Now, they optimize differentiation protocols in a 2D format, high throughput format, and what their customers are interested to know is then how do they scale up from implementing this differentiation in 2D. And we also worked in this collaboration with Imperial College, Hugh Brady's group, who have a lot of experience in extended flow marker panel design. And they really contributed to the assessing the quality of the product. So this is a 35-day process, manual process in 2D, which, as you can see at the bottom, quite typical of actually an academic process. And what we worked on was to make this entirely a 3D STR-based process. It went very well. So we had achieved a 10x yield improvement in the expansion phase and the diff phase of the NK cells. So 10x yield on the final NK cells exiting the process. Now, when we looked at the quality side of things, we looked at three critical process parameter combinations, three experimental conditions, if you like, and assessed the cell quality initially with a basic marker panel, which many people use for NK cells. And you can see with the orange bars that the outcome was fairly, they looked about the same, and nothing to worry about there, really. But when we plotted our functional assay results against the extended marker panel results, we found that, in fact, there are differences in the outcome populations. Some of the populations were more cytotoxic and some were more productive. And what we found was that we should be optimizing for both of those parameters. Now, if your process conditions or changes in your process conditions can have such a pronounced effect on the quality of the product, and the characterization of this quality is so complex, there is a bit of a gap. We need to understand this more, and what's more, we need to find a way to monitor this in order to control it within the bioreactor through some easy assay at line or online in the future. So we looked further into in-depth characterization. We looked at transcriptomic analysis, proteomic analysis, also microRNA and metabolomic analysis, but I will not show you those two today. You can see, so from single-cell RNA-seq data of the three process conditions, quite a pronounced difference in the cell populations here, where condition C is drastically different in terms of the cell populations and far higher in NK cell population. Now, how do we track this? How do you find how to track this throughout the culture? This is where we looked at the targeted protein screening. So we did a protein screen using the Olink platform, which is a high throughput screening platform. We looked at 5,400 proteins within this screen. And on the PCA analysis here, you can tell that we can see a difference in the proteome, and we can potentially then go back and link to identify some of the proteins that we should be monitoring throughout the process. because we can track this. Now, this is kind of where we're at the moment, but this is absolutely a gap and an opportunity for assay development or for sensor development for the future. These kind of data sets are laborious and very difficult to obtain. They take a lot of time, and you need to generate high throughput experimental conditions in order to screen those process parameters. And we are now talking about an extended characterization for QC analysis. So what we've done is to create what we call a patent biomarker discovery laboratory, which is an automated setup for development and characterization. It's a lab which does automated sampling from high throughput bioreactor systems, and that automated sampling rig is connected to at-line as well as a liquid handler platform. At-line measurements for things you might expect to measure, cell and metabolite analysis. And all of this infrastructure is connected to a data collection system for structured databasing. Now, what this allows you to do is to use this data now to create, to put it to work, essentially to create a digital twin of your process, which can then help to control your process either live, so automate feeding and control, or to help inform future experiments. So the platform we have integrated is from SecureCell. It is a Numera automated sampler connected to the rest of our systems, and as the software is called, Lucullus software. So we currently have two. Well, actually, there is a third. We have 3 digital twin projects ongoing at the moment. 2 of those are in the AAV space, and 1 is looking at TILs for autologous application. And this is the idea that we should be able to apply and hope to apply to the IPS space in particular, where we will be able to further control and take out the manual steps from the process control within our SDR. Now, we've gone over the IPS case study. So we looked at the process closure within the SDR environment and taking the manual steps out, how to maximize yield and quality within this SDR, and how to get onto the path of a PAT for control. Switching gears a little bit now into scaling for scale out application, such as CAR-T or others. You have seen the process flow, but just to give you an opportunity, we're now talking about going from patient back to the patient, so taking blood from the patient, delivering it to a manufacturing site, starting up a manufacturing process, which is 3 to 12 days long, QC testing, batch release, and then back to patient. And this material, starting material and product material, can be a mix of fresh or frozen, as therapy developers take different approaches here. The sorts of batch sizes we were talking about are very, very high in theory. And so the approach we have taken is to really try to simplify, rather than, you have seen the IPS example, which is highly complex. Here we try to remove as many steps as possible. And we have used the G-Rex platform for this. It's quite popular now for among therapy developers because of the simplicity. It is essentially a flask where cells can sit on top of a membrane for oxygenation. And this flask will keep the cell culture throughout that 12-day process, and there'll be some additions of feeds, transduction made to it. So the steps here we'll talk about are minimize intervention and maximize reproducibility as much as you can. One of the challenges and the first area we saw that needed tackling is the controlled parallelization, and particularly within the expansion time period of the step. This is the first step that therapy developers look at. There's also a challenge in QC automation. We have made some steps towards this, and there is real need for digital integration and advanced control. The gap, a key gap here, as many of you work for companies that are working on technologies within the space, there's so much innovation, so much opportunity, and the gap here is in moving towards deployment and GMP deployment. And this is a challenge as for therapy developers and device developers alike. Therapy developers struggle with building the case for this, demonstrating the value, understanding when automation is required, and managing a fairly limited budget as it is now. With new devices, there is a point where trust needs to be built. People need to understand what's required for implementation, how they will put this to use, and what this project plan will look like. So we have broken this down into 3 stages and hope to facilitate the building of this value chain over time, so that it is de-risking the work that the therapy developers perceive it to be. We start with a strategic assessment of feasibility analysis before testing and deployment. Our first project in collaboration is with Cellular Origins. So we now have the Cellular Origins robot, which is shown here, which has arrived into our clean rooms, and we're beginning to work with this to implement a CAR-T exemplar process. Now, on the QC automation side, there are several challenges that we see need to be addressed. We currently, in the cell and gene therapy space, rely on multiple offline instruments, and the QC footprint and requirement is really quite big. There are inherent variability of the living samples that we are putting into this QC techniques, and they are very manual, and there is a lack of standardization in some of the methods that we are using. So there is real opportunity and need for automation and digitalization within this space. We need increased throughput, improved quality, and reduced variability, and overall complexity. So as much as we can condense the analytics into fewer techniques that give us the most output, we can then work to automate those, and it will be far more worthwhile. This is an example of where we have automated a qPCR workflow, where our team had initially thought that we couldn't move to qPCR because of the reproducibility issues that we were seeing with the technique, that we were using digital droplet PCR at the time, but through automation we actually have achieved the same output of the assay, so the quality of the assay improved to make the automation worthwhile, and then we had cut the costs pretty much in half. So as I mentioned, there is so much innovation within the space, and so much need from the therapy developer side. So we have established an ATMP specific GMP mirroring test beds. So these are GMP-like clean rooms in the UK, in Stevenage, which can host these automation technologies and digital technologies to enable the collaboration needed and the connections needed to be made between unit operations and between systems. The clean rooms are unique in that they do not come under stringency of GMP, a GMP environment, but they are built as GMP clean rooms. So as you bring your automation kit in there, particularly if it's big and bulky and large automation, you can start in a non-GMP state, build it up, and then flip the clean room into a GMP state, as we have already all the systems for GMP in place. It's just a question of shutting one of the exits off and rerouting into GMP entrance. With the Cellular Origins collaboration, we are first addressing the expansion part of the CAR-T flow. The figures that they hope to achieve are shown here, and I just hope that we get there. It's a very ambitious target. But we will see, and this is part of where I think cell therapy developers really are interested to see what is the reality of automation rather than the ambitious figures, the potential figures in the future. What can you really achieve with once the solutions are in place, they are validated and under all of the restrictions that will be applied to it. So this is the collaborations that we have had over the years in the iPSC space and the cell space. We have had more than 20 collaborations within this and have broad experience across cell therapy modalities, and now are really working on this gap between innovation and deployment. I wanted to say a huge thank you to everyone involved in the work that I have shown today, from the Catapult, Plasticell, Imperial College, and there was also some work from the Earlham Institute. And thank you to you for listening.